Hi everyone. This week we're on microbial growth and nutrition part 2. And in section 6.3, we're going to be talking about how bacterial cells divide. You know a little bit about cell division. You learned it in another class. You might remember learning about mitosis. And we talked about mitosis in the context of eukaryotic cells because eukaryotic cells have many linear chromosomes. Remember, human cells have 46 linear chromosomes in the nucleus. And before that cell divides, those chromosomes need to be sorted out so the correct 46 go to one cell and the correct 46 go to another cell. So, and bacteria are a little bit different. Prokaryotes are different because prokaryotes don't have to do all that sorting of chromosomes. So you don't need the complexities of all the stages of mitosis. So bacterial cells have a simpler method of dividing, and we call that binary fission. And in this diagram, what it's showing you is just, if you start with one bacterial cell, it's going to divide into two equal cells. And that's how your cells are going to divide. You're going to have one divide into two, and then two will divide into four, et cetera, et cetera. And here's just a little diagram of binary fission, which is much simpler than mitosis, so that you don't have all the steps to learn. But remember, bacterial cells have a single circular chromosome, and the first thing that has to happen is that chromosome has to replicate. That you have to make a copy of the DNA, and then that cell, see how the cell's kind of getting larger? It's going to pinch off into two, and one copy of the DNA is going to go to one daughter cell, and the other will go to another daughter cell. And I'm not going to give you much more detail than that, but the idea is when bacterial cells are grown, we start with one cell, we end up with two cells that are identical to the parent. So when we're talking about growth, and we mentioned this in part 1 of microbial growth, we're not talking about how one cell grows. We're not talking about like human development and over the years. When we're talking about bacterial growth, we're talking about the growth of populations. Because when a bacterial cell reproduces, you have one cell, you get two cells that are just like the original. There's no like fancy development going on there. So when we're talking about bacterial growth, we're talking about the growth of the population. And so when we're looking at the population or the actual number of cells in total, we use some of the vocabulary. We use this word doubling time. Doubling time is also called generation time. It means the same thing. So you should recognize both of those words. So define doubling time or generation time and explain how it leads to exponential growth. Well, so what the heck is exponential? Well, actually, we'll back up. I'll come to exponential growth in a minute. But what we're talking about with doubling time is the amount of time it takes a bacterial population to double in numbers, or the generation time, the amount of time it takes for a population to double in numbers. So if you look at E. coli is a really common bacterium. We all carry it in our intestines. Although, there's lots of other kinds of bacteria, but one of the reasons why E. coli has become kind of one of the gold standards for studying microbiology is, like I said, we all have it with us. There's lots of kinds of E. coli that are not pathogenic. But one of the big things is it's got a really short generation time, a really short doubling time. So for a population of E. coli to double in number, depending on the conditions and the strain of E. coli, it can take anywhere from about 20 minutes to an hour. And so that would be relatively quickly for a number of bacterial cells to double in 20 to 60 minutes. There's other variations of that. If you look at this bacterium called Mycobacterium tuberculosis, this is the bacteria that causes tuberculosis, the really bad respiratory disease. The doubling time for Mycobacterium tuberculosis is 15 to 20 hours. So when I was in graduate school, I worked in a lab with a kind of bacteria. We used bacteria that had a doubling time around E. coli, like 20, 30-minute doubling time. The folks down the hall, they grew Mycobacterium. They studied Mycobacterium. I could start my cultures I could start my bacteria at the end of the day, come back the next day, and I could do my experiments. I'd have lots of bacteria. The mycobacterium folks, they had to start their bacterial cultures and then wait about 2 weeks before they had enough bacteria to do their cultures. And so it's just inherent. It's just something about the way these different microorganisms grow that the generation times can vary tremendously. If we look at this picture, what you see is here we have it's generation zero. We starting with one cell. After the first generation, the cells have doubled. So one cell divided into two by binary fission. The next generation, each of those cells is going to divide into two. So I went from 2 cells to 4 cells. I get to the next generation, and those 4 cells are going to divide into 8 cells. And so what we're going to see as bacteria populations grow is the more bacteria I start with, the bigger the population is going to get faster because I'm going to double at every generation. And this is why we say bacterial populations grow by exponential growth, or we say they grow exponentially, meaning the larger the population is, the faster it will grow. You get that? The larger the population is, the faster it will grow. If I start off, it's going to take the same amount of time for one cell to make 2 as it's going to take 1 million cells to make 2 million cells. But if I start off with a high number, if I start off with 1 million, my numbers are going to get higher faster. So we say bacteria grow exponentially. Their populations grow exponentially. Okay. So when we're looking at these populations, it says we're going to kind of look at the way they grow. Our learning outcome is calculate the number of cells present for a culture with a specific doubling time or specific generation time. So here's a problem you should be able to do. Here it says, how many cells would be in a colony of bacteria that has a generation time of 30 minutes if the plate was incubated for 6 hours? Look at that question carefully. See if you can figure it out. Every generation, you're going to double in numbers. How many cells would be in a colony of bacteria that has a generation time of 30 minutes if the plate was incubated for 6 hours? See if you can figure that out. The first thing you're going to need to know is how many generations do I have? Well, let's see. I have it doubles, or one generation is 30 minutes, but we're going to incubate for 6 hours. So that means I have 12 generations. If I start with one bacterial cell, after one generation, I'm going to have 2. After 2 generations, I'm going to have 4. After 3 generations, I'm going to have 8, and on and on and on. And that's good. You could do that. We could keep going. So 8, 16, 32, 64, 128, and you'll be able to figure this out. But it's easy to make a mistake. So there actually is an easier way to calculate this. If you take 2 to the exponent of the number of generations and you plug that into your calculator, there's your answer. So I could go 1 to 2 to 4 to 8 to 16 to 32 to 64 to 128 and keep going and going. And after 12 generations, I'd get to 4,096. Or I could do 2 to the given an exponent of the number of generations, and that would also give me the same number. So that's a way we can calculate that. Okay, so do the same problem again. What if we let the plate incubate for 24 hours? So if we're going to start with one cell, the generation time's 30 minutes, and we're going to incubate it for 24 hours. So let's see, 24 hours, 30-minute generation time. That means 48 generations. So if I go 2 to the 48, Oh my gosh. What is that one? A million, a billion, trillion. You'd have 281 trillion cells if we took one bacterial cell that had a generation time of 30 minutes and we incubated it for 24 hours. We'd have a trillion cells. When I do the calculations, you're going to do this in lab actually. When you do the calculations on a colony, we usually get in the hundreds of millions and not the trillions. So the thing you have to think about microbial growth is something's going to happen and the bacteria are going to start running out of nutrients or they're going to accumulate a lot of waste, things like that. So there are things that stop bacterial growth. And they will not necessarily grow indefinitely. But you can at least get an appreciation for the how fast and how many bacterial cells you can get in a relatively short period of time. The last thing I want to talk about in relation to microbial growth is in relation to bacterial growth is what we call a bacterial growth curve. So our learning outcome is describe what is happening in the bacterial population during the 4 phases of growth in a bacterial growth curve. Now, this is something that we can do in the laboratory, and we do it for experimental reasons for all different things, but bacteria grow this way in natural populations too. So the natural way a population of bacteria grows, we call this a growth curve. What did I just do? Okay, so before I show you the growth curve, before I show you the stage of the growth curve, I want you to notice that here's a graph, and this says logarithmic of living bacterial cells. So this is essentially the number of cells versus time. And when I use logarithms when we're talking about that exponential growth. When the bigger the number I start with, the faster you're going to get to a higher number. Here is a graph that just has time versus number of cells. And what you see is when I go from 1 cell to 2 cells and 2 to 4 and 4 to 8 and 8 to 16, it looks like I start growing kind of slowly at first. And then when I get to 100 to 200 to 400 to 800, from 1 million to 2 million to 4 million, it's going to look like the bacteria are growing faster and faster if I plot that growth on a linear scale. If I put this on just a linear scale, like from 1 to 2 to 3 to 4 to 5. But the bacteria are actually growing at a constant rate. When you're growing logarithmically, when it takes the same amount of time to go from 1 cell to 2 cells as it takes to go from 1 million to 2 million, like I said, we call that exponential growth. And we graph that on what we call semi-log paper. So instead of having a linear scale on this y-axis, we have a logarithmic scale. It doesn't matter that you completely understand logarithms or not. I know probably most of you have some experience with that with the math class. What the bottom line is when we graph exponential growth using a logarithmic scale, a straight line is going to mean I have a constant growth rate. That's what I want you to know. Okay, so when we plot the growth of bacterial populations, here it says logarithm of living bacterial cell number. That means it's plotted, so a straight line is going to represent a constant growth rate. So here we have, when we do a growth curve, we're going to plot the number of cells versus time. When we look at what we call a bacterial growth curve, what you'll typically see are 4 stages. So here we have the number of bacterial cells in the population versus time. And you'll notice when you start the culture, and this could be, like a bacteria ends up in a broth or on an agar plate, or it could be bacteria end up in a wound or in your blood. but the bacteria frequently go through something we call lag phase. So what this is showing you is the number of bacteria are not increasing. We have a short period of time where the number of bacteria or the number of the bacteria in the population are not increasing. They're lagging. There's no increase in the number of cells, but lag phase is actually very metabolically active. The cells are actually getting ready to divide. They're copying their DNA. They're cranking up their metabolisms, and they're getting ready to divide. These are actively living, metabolizing cells. So eventually they've got, enough proteins made. They've got their DNA copied, and then they start growing logarithmically. We also call that exponential growth. That's where the number of the cells are doubling. We're going from whatever number we started with, 10 to 20 to 40 to 80 to 180, 160 to 220 or 320, so we're going on and on and on. So the second stage of bacterial growth is the log phase. And this is where bacteria are going to grow very quickly. And that's where your doubling is going to happen. That's where you would calculate your generation time, your doubling time. So we start, if we start it in a broth, we put our bacteria in the broth. They got used to the broth, they got metabolically ready, and then they started doubling. And so you can see this constant rate of growth. Now, and so we call that log phase. So we have lag phase and log phase. Log for logarithmic, also meaning exponential. It shouldn't be surprising that bacteria won't grow forever. If bacteria grew forever, our planet would be covered with miles and miles of bacteria. But there's things that stop bacterial growth. And so what you can see here is in a normal bacterial growth pattern, you're going to grow to a point, and then you're going to kind of level off into the stationary phase. So something has to happen to kick this population into stationary phase to stop it from growing. Actually, in stationary phase, you don't really stop growing. For every cell, that your environment is just good enough to support the population that's there. So for every cell that divides, another cell has to die. So you're supporting a stable number of cells in that population. Something happens at the beginning of this stationary phase or the end of log phase that puts stress on the population. The bacteria might be running out of food. They might be running out of oxygen. They might be making too much waste. And that causes the culture to go into stationary phase. Eventually, the conditions get to a point that the environment can't support the bacteria anymore, and we'll get a death rate that exceeds the growth rate, and that's when you're in death or decline stage. The important thing there is death also occurs exponentially. And so death rate is going to happen gradually or logarithmically, just like growth rate. This is really important. Like when you're talking about somebody has an infection and you're trying to, and the infection is the bacteria are dying. Let's say your immune system's fighting it off and the bacteria are dying, or you're taking a drug and the bacteria are dying, something like that. To know that the whole population isn't likely to just die all at once. They're going to die over time and gradually. So the 4 stages of growth curve: lag, metabolically active but not dividing; log, where you can calculate your generation time, and bacteria growing their fastest. At the end of log stage, environmental conditions happen, so to make it not being able to support any more bacteria, so you go into stationary, where the number of cells dying and growing are the same, and eventually conditions deteriorate to the point that death rate's going to exceed growth rate. That's the bacterial growth curve.